Good morning, students. In today's class, we are going to continue the topic, that is how do organisms reproduce. In last class, I gave you some basic introduction and we stepped into certain concepts of that lesson. So once again, let us have a small recap of the things that what we discussed in the previous session. Yes, so now you have some idea of what this term is, reproduction. Yes, that is re, which means what? Again, and production, that is to make. So, reproduction is a process of making or creating a new organism of its own kind. Why this reproduction is very important? What for an organism should reproduce? Reproduction is a very important process that makes the organism to survive in this world. What is called survival? Survival in the sense an organism that should cope up or that should possess the ability to withstand all the biotic and abiotic stresses. If such an organism do that all, then that organism can sustain and they can have their own population and they can persist in the world. Yes. So reproduction is a very important process for making an organism to survive. Yes. So then this reproduction is of two types, that is asexual and sexual. Later we shall discuss the difference between what is sexual and asexual. Now we are going to see a very important and the interesting topic here, that is what happens during reproduction. So before that, let us have a small recap the things that what we discussed in 9th standard, the lesson cell. Yes. So in that, I guess you remember that what this structure is called the chromosome. Yes, so this is the structure of chromosome, and what this area is the condensed form of your DNA. Yes, and if you maximize this area, then what you get here is the double stranded form of DNA. So this is the organization of chromosome in human being. So all the organism, they possess several set of genes, and only with that genes they make their structure. Now my question is, how this gene is giving structure to the organism? Say for example, you are a human being and myself a human being. Yes, but still, though we are a human being, why we have some variations? Because you have certain different gene in you and I have certain different gene with me, apart from the common genes that what we possess. Once again telling you, what is the variation is, the difference between you and me is called the variation. Yes, simply in biologically speaking, variation is defined as the degree of difference among the organism. That is the variation. Yes. So this variation is purely based on structural difference of an organism. It is not necessary that always structure should vary. Even sometimes the organism may appear to be similar, but genetically they may not be similar. Yes. So this DNA, and last year I remember that we studied the cell lesson. At the time we were discussing about the protein production. Yes. What is protein? Protein is a molecule that brings structure. Yes, protein is a product of gene, and protein is produced by several biological process, and that gives you the protein. Okay, so now it's very clear that gene makes the protein and protein gives the structure. Once again telling you, gene makes the protein and this protein is giving structure to the organism. So if a gene gets changed, then protein alters. If protein alters, then structure may change. Let me explain you in Tamil. Gene அப்படிங்கிறது தான் புரோட்டீனை ப்ரொடியூஸ் பண்ணியிருக்கு. எஸ், இந்த புரோட்டீன் அப்படிங்கிறது தான் ஒரு ஸ்ட்ரக்சர், ஒரு என்ன சொல்றது, ஹைட்டு, வெயிட், த கேரக்டர்ஸ், வாட் ஆர் த திங்ஸ் வி ஆர் சீயிங் இஸ் த ஸ்ட்ரக்சர். எஸ், ஸோ அப்போ ஜீன் சேஞ்ச் ஆச்சுன்னா புரோட்டீன் சேஞ்ச் ஆகும், புரோட்டீன் சேஞ்ச் ஆச்சுன்னா ஸ்ட்ரக்சர் சேஞ்ச் ஆகுது, ஸ்ட்ரக்சர் சேஞ்ச் ஆச்சு அப்படின்னா வேரியேஷன் அதிகமாக வருது. எஸ், ஸோ விச் மீன்ஸ் வாட் ஹியர் இஸ் வெரி இம்பார்ட்டன்ட் டு ஹாவ் த வேரியேஷன் இஸ் change in gene. So change in gene give rise to change in protein, and change in protein give rise to change in structure, and change in structure gives variation. Yes, so this is what we are going to see. So which means now here gene is a part of DNA where the information is stored. How you can define this gene? Gene is a piece of, or it is a part of DNA that has the information stored to make the particular protein. Yes, so if this gene gets altered, okay, if the gene gets altered, the changes you can see in protein. Once the protein gets changed, obviously the structure of the organism start to change, and if the difference is more, then the variation obviously it's going to be more. Yes, so that makes an organism that may not be similar to the next organism. Okay, fine. Now what for this variation to occur? Why? What is the necessity for an organism to focus on this variation? Say for example, in the theory of evolution, there must be one organism that was the first organism that might have emerged. Yes, see, evolutionary theories are all the imagination. Okay, like we don't have any proof, but we should believe that. Yes, so likewise, and say for example, if bacteria, just assume that if bacteria is the first organism that evolved in the evolution, if bacteria doesn't show any variations, which means what? If the gene present in the bacteria remains constant, then still the bacteria may keep dividing and only the bacterial population might have survived now. Yes, but this bacteria slowly they started accumulating variation, and from bacteria maybe the protozoa might have emerged, and from protozoa maybe some new organism, the next to that cadre might have emerged, and at last the product that what we are the human beings that we are raised from these tiny organisms. So variation is a key factor for bringing evolutionary changes. That is point number one. And one more thing the variation do, what I told you, the first is variation brings the evolution process to run up very fast. Yes, the second thing is variation brings adaptiveness. What is meant by adaptiveness? Adaptation. That is, say for example, if this is the bacteria, they are living in one normal habitat. You know what is meant by habitat? The place where an organism living is called habitat. Yes, so assume that this bacteria has got its own adaptivity and they are living in a certain habitat. Now due to global warming, you know what is global warming? That is gradual increasing in the Earth's temperature is called the global warming. Due to this impact, now this habitat of this bacteria, they are going to experience certain rise in temperature. So most of the bacteria they start to die. Why? Because they don't have any adaptive features. So some bacteria they may become resistant. This resistant is a different version of this mother bacteria. Let me explain you once again. See here, this is the bacteria and they have already been adapted and habituated to a particular habitat, and due to the impact of global warming, the particular bacteria now it's going to experience a new environment where the temperature is drastically increasing. But normally this bacteria they don't have any heat tolerance, but certain bacteria now they change their gene here and slowly they are getting adapted to the particular fluctuation in the temperature. So that adaptation is possible only because of this changing in the gene, and that is called the variation. Yes. So now it's very clear that variation leads two way. One is it has its own survival advantage. Yes, here you see, as the heat is going to kill the bacteria, and those bacteria, those have the capacity to induce variation, only that bacteria is surviving. Yes. So that is one case with the variation. The other one is this variation brings more changes, so they are making a new organism from pre-existing organism, that is otherwise called as evolution. Yes. So now it's very clear that gene plays a very important role in maintaining the organism and making a process of evolution, and it's bringing even survival advantage. This gene should be inherited from parents to offspring. Yes, that is called DNA copying. Yes. Now the next topic what we are going to see is DNA copying. Yes, so now it's very clear that DNA, that is what? DNA, the full form for DNA is deoxyribonucleic acid, and they have several genes inside, that is simply called as genome. Yes, say for example, we have 23 pairs of chromosomes and we have billions and billions of genes that present in each chromosome. Yes, together the overall length of an DNA of an organism and a cell is called the genome. Genome contains several genes, and gene is a piece of DNA. Again telling you, genome contains many genes, and gene is a fragment of DNA. Yes, now I guess it's very clear for you, right? So this DNA should get copied from the mother organism to the daughter organism. I mean what? That is a progeny or offspring. So it's very important that this DNA, that is a gene, gives structure and several what? Cellular process. But this DNA copying may not be accurate. Why? Why DNA copying is not accurate? You know the reason why? Because you see, it is not simply a chemical reaction that we can predict. In chemistry, we can predict based on seeing the reactant, we can tell instantly what the product would be formed as the end of the reaction. But in biology, it is not possible. Why? Because it is a complex biochemical reaction. Complex biochemical reaction, which means it is not a one-step process. It takes several processes, and it's like a cascadal reaction. You know what is meant by cascadal reaction? That is, it's not like, say for example, hydrogen combines with oxygen to form water. No, you can't tell anything such kind of reaction exists in biology. See, for converting a glucose into pyruvate, it is like a several process that takes us intermediate steps. So in your higher classes, you will be studying that the particular step for converting the glucose into pyruvate, that is called glycolysis. Yes. So likewise, it is a complex process. So as the number of step increases, the accuracy decreases. So you can't tell always that the DNA copying is exact. The copy produced by the mother cell will never be identical to the new cell. Yes. So that is the main reason this variation is what is emerging out. Is but DNA copying brings more variations. That is what the concept we learned from here. So do organisms create exact copies of themselves? Do organisms create exact copies themselves? No. Say for example, father mother. Yes, assume that we are going to study what happening here. Father, he has some characters, the uniqueness and the novel characters, and mother, she possesses some unique characters. And during the course of reproduction, the son or the daughter produced will have the combination of both father as well as the mother. Yes. So which means father has donated his own gene to his son, and mother obviously is donating her own genes to the son. But son possesses both the character of father as well as mother. Not only that character, and some unique characters which is not being seen with father and mother. Such kind of new characters that every organism they possess. Yes, and sometimes the characters may get skipped from father and maybe receiving from the grandfathers or grandparents. Yes, so this is called inheritance. This we are going to study in the next chapter, that why this is all happening. So in order to have this all process held, DNA copying is very important. Yes, so the organism, that whatever the copies they are making themselves, will never be identical to each other. Only because of that we could experience more variation. More the variation occur, more chance that the new organism can emerge out from pre-existing population. That is simply called as evolution. So now it's very clear that DNA copying is very essential. What for? To get more vital gene for the next the offsprings from the parental traits. Yes. The next importance of variation that just now we studied. The two important, one is to sustain the evolutionary, what that sustain the stresses, what the abiotic and biotic form we are getting, and second one is to proceed with the process of evolution. Is next mode of reproduction. Mode of reproduction. Yes, already we discussed two modes they exist. One is asexual, the other one is sexual. As we know here that asexual, they involve how many parents? Only one parent. And sexual, they involve two parents of opposite sex. And the product or the offspring that produced as a result of asexual reproduction are genetically identical. You know what is meant by genetically identical? If you check the gene of the offspring with the gene of the parent from where the offspring are produced, both will be same. Simply, it is called as clone. Clone, C-L-O-N-E. C-L-O-N-E. Clone. Clone is, say for example, if this is the organism, is if this organism produces, if this is the organism, and this organism, they are going to divide into two equal. And you see these are the offsprings. Yes, and this is the mother. So now it's very clear that this organism is making exact Xerox copy of the same kind. Is so now it's a best example for asexual reproduction. So what the character this mother possesses, obviously both the offspring they possess the same character. Why? Because it is making exact of its Xerox copying. So DNA copying occurred here is exact similar to its mother, but it is not necessary that it should be identical. Yes, slight variations may happen in asexual reproduction. Again telling you students, asexual reproduction produces more clone. It doesn't mean that they don't possess any variation. They do possess variation, but the degree of variation is very less, or at times it may not be noticeable. Yes, such kind of variation that exists. You know, one more term I thought of introducing here, that is called mutation. What is mutation? Mutation is any alteration or any changes where the gene gets altered. Only then, if the gene gets altered, then it can be expressed as an altered protein. That altered protein is going to alter the structure of an organism. Yes. So listen here again. See, mutation. How you can define this mutation? Mutation is change in DNA. Here DNA represents gene. Change in DNA, and as a result of mutation, as a result of mutation, we know that mutation is change in the gene. So as a result of mutation, change in protein occur. Yes, as a result of change in protein, change in structure occur. Yes, as a result of change in structure, they start possessing more variation. So now my question is, what brings variation to an organism? It is now very clear that mutation brings variation. Mutation is the change in DNA. Once the DNA or the gene gets changed, then change in protein starts occurring. As we know that protein is going to determine the structure. Yes. So if the protein changes, obviously what will change? Protein மாறிடுச்சு. அப்ப என்ன மாறும்? ஸ்ட்ரக்சரும் மாறப்போகுது. ஸ்ட்ரக்சர் மாறுது அப்படின்னா என்ன ஆகும் அடுத்து? வேரியேஷன் அதிகமாக வரும். அப்போ if an organism starts showing more mutation, there is a high chance of more variation to occur. Yes? So this mutation is of two types. Okay. One is beneficial mutation, the other one is lethal mutation. What is meant by beneficial? By the name itself you can tell. Beneficial in the sense, yes, some genes, because of the alteration, so they are going to make you or make an organism get benefited, which means like here we stated about adaptations, right? So if certain organism, they are not adapted to the heat, and some organism due to the mutation, they are getting adapted to the hot environment, right? So such kind of mutation is called beneficial mutation. So as a result of beneficial mutation, the organism, they are acquiring certain good characters or good changes. Such kind of mutation is called beneficial mutation. And some mutation, they are undesired by the organism. Undesired அப்படினது தேவையே படாது அந்த mutation. A best example is what? The best example is cancer. Cancer, how cancer occur? Cancer is all due to the mutation that occur in certain cells, and they are breaking up the what? Cell cycle, and they start proliferating, and they start in an uncontrollable manner. Yes. So such kind of mutation, they bring death to one organism. Such kind of mutation here is called lethal mutation. Again telling you, mutation is the change in DNA that is carried to the protein. The change what the gene has acquired, that is going to get reflected in the protein. Once the protein changes, the structure of an organism changes. Once the structure of an organism changes, then obviously it's going to have more variation with among the population. Yes. And this mutation is of two types. One is the beneficial mutation, the other one is the lethal mutation. Beneficial mutation is going to bring more important survival advantage, or whatever the thing the organism is going to get benefited. But in case of the lethal mutation, here lethal is dead. Yes. So this mutation is unwanted or nuisance mutation, that it's going to cause the organism to die immediately, or that is the terminal thing that happens for an organism to get wiped off from the evolution. Yes. That's all about the mutation here. Now we were discussing about the mode of reproduction, that is asexual and sexual mode. Yes, let me make a small diagram here. Fish, You can understand by seeing the diagram. Yes. So now you see here, this is for asexual reproduction. Asexual reproduction, and here is for sexual reproduction. Yes. Say for example, now assume that this is one organism that it's going to reproduce asexually, and here for making a sexual reproduction to occur, how many parents are required? Two. Let me make a small differentiation here. So this is organism number 1 and this is organism number 2. Yes. So here in case of sexual reproduction, we know that the organism or the offspring produced as a result of asexual reproduction will be identical to its parent, simply called as clonal method of reproduction. Is here too DNA copying has occurred, but the DNA is exactly copied. It is not exactly, but it is like if you want to tell it in a percentage, it's like 99.9 or something close, which means what? It is not the exact copy, but it looks more similar in terms. Yes. So now the parental organism has produced two exact copies of the organism, and this parent is genetically identical to the offspring what it is produced, which means they don't possess any variations. Yes, but the variation is very limited. Again and again telling you students, let me tell you in Tamil, அதாவது வேரியேஷன் இல்லை அப்படின்னு நம்ம சொல்லலை. ஏசெக்ஷுவல்ல வேரியேஷன் இருக்கும், பட் அது நோட்டீஸ் பண்ண முடியாத அளவுக்கு ரொம்ப மைல்டா இருக்கும். புரிஞ்சா? பட் here in case of sexual reproduction, the variation is very clear and prominent. Prominent in the sense what? It is more visible clearly. Yes. So now it's a sexual reproduction part we are going to see, and these organisms, they produce their own gametes, that is germ cell. Just assume that this is male and this is female. Yes, and the sign for a male is, it looks like sperm cells, and female sign you need to make it like this. Why? It is like an anchor, which means it can't move from place. We know that sperms are motile and eggs are non-motile in nature. So in order to understand biologically, you can represent the male and the female organism in this manner. Yes. So assume that this is a male organism and this is the female organism, that they are going to make a new organism. So first step of a sexual reproduction is gamete formation. This gamete is the sperm and ova. So now as this is the male organism, they are producing their own kind of sperm cells, and now this is the egg cell. Yes. And now you see here, the 50% of the DNA of this organism goes to this sperm cell. So listen here, if this is one cell, the germ cell produced as a result of this meiosis that you studied in your 9th, right? So meiosis produces germ cells in testes and ovaries, isn't it? So in that case, you see 50% of the parental cell, the DNA will be inherited or passed to the next, the sperm cells here via, and the other 50% goes to the another sperm cells, and the two sperms produced as from the one mother cell will never be identical. Yes, here it is identical, here it is not identical, and moreover here if it possesses 100% DNA, okay, here in this case once again it is making the 100%, which means what? Exactly it is giving a Xerox copy of its own. So obviously there will not be any variation. Yes, but here you see, if this is the 100%, okay, the sperm carries 50% of the DNA in its own. புரிஞ்சுக்க முடியுதா நான் என்ன சொல்றேன்னு? இப்போ இதுல 100% DNA இருக்கு. இந்த 100% DNAல 50% ஒரு ஸ்பெர்ம் செல்லுக்கும் இன்னொரு 50% இன்னொரு ஸ்பெர்ம் செல்லுக்கும் போயிடும். அப்போ இந்த 50யும் இந்த 50யும் ஐடென்டிக்கல் கிடையாது. ஏன்னா இதுல பாதி இருக்கு, இதுல மீதி இருக்கு. எஸ், லைக்வைஸ் here in case of ova, again it's 50%. From this 100% it goes to one ova, and again here the 50% from 100 it shares the equal amount. Now as a result of fertilization, we know that sperm fuses with the ova. Yes, so this 50% from sperm and 50% from ova makes again 100%, but that is a combination of the partial from the father and partial from the mother. So the offspring produced, once again they start possessing the same 100% like their parents, but they are the combination of parents, which means here both the characters, they may get blend or they may get contrasted, or a new character may get what expressed. Oh yes. So again here you see it's 100%, and you see again I told you this is not equal to this and this is not equal to this. So which means the offspring produced by the same parents, the offspring also may not be equal. That is the reason you see if you have a sibling and you and your sibling have a same parent, but you may not be looking alike with each other, but you have more resemblance. Yes. So it is all because the sexual reproduction brings more variation. Yes, here it brings very less. You can't tell that it will never bring any variation. Why? Because DNA copying is not always accurate. Again telling you students, it's a very important question you can expect. Why DNA copying is not accurate? The reason is DNA copying is a biochemical reaction. As we know that biochemical reaction, it is not always that you can expect the reaction to happen as per your predictions. Yes. So there may be some error. That error that happens during that reaction is technically called as mutations. நல்லா தெரிஞ்சுக்கோ, டிஎன்ஏ காப்பிங் அப்ப மியூட்டேஷன் நடக்க தான் செய்யும். அந்த மியூட்டேஷன் நடந்தாதான் சேஞ்சஸ் வரும். அந்த சேஞ்சஸ் வந்தாதான் வேரியேஷன் வர முடியும். அந்த வேரியேஷன் வந்தாதான் அடாப்டேஷன்ஸ் வர முடியும், எவல்யூஷனும் நடக்க முடியும். That is the main reason. Again and again I was telling you what is DNA copying and why DNA copying is not accurate, and again in which reproduction this variation is focused more. So now it's very clear for you to understand asexual reproduction is not preferred by more organism. Why? Because it is showing very less variation. But sexual reproduction is highly preferred by many organisms. But if you compare that with unicellular and multicellular, most of the unicellular they follow asexual reproduction. Why? Because you see this reproduction is otherwise called as growth rate. Yes. If you put this as a yeast, ஒரு yeast cell அப்படின்னு நீ எடுத்துக்கிட்ட அப்படின்னா அந்த yeast அப்படியே பட் ஆயிட்டே போயிட்டே இருக்கு, அதை நீ growthனு சொல்லுவேன். But here in this case you see, you can tell that it's even doing its own reproduction, isn't it? So reproduction can be equated with growth in case of asexual reproduction. But here you see these organisms, they have a various anatomical difference that supports the sexual reproduction process to happen. For that they should possess first a germ cell. Yes, that is the main reason. For any organism that participate in sexual reproduction, that should possess a sex organ. In case of animals it is called as testis and ovaries. In case of plants it is called as simply called as andrecium and gynecium. That all we will be seeing in the next forthcoming classes. Yes. So I hope now it's very clear for you what is the basic difference between sexual and asexual reproduction. Yes. So now we are going to see about various modes that occur in asexual reproduction. Let us make it one by one. That is, now the first process here is the fission. Fission in the sense what? That is breaking or splitting. Yes, which means what? It exactly breaks or it exactly divides into two equal halves, not two or many pieces. Yes. So this fission, based on number of cells produced as a product of fission, it can be again divided into two. Again telling you students, fission அப்படினா பிஞ்சு போறது. Yes, which means it's breaking into many or some few. Yes, based on how many cells are produced. If it is 2, as a result of fission, if 2 daughter cells are produced, then it is called binary fission. Yes, we know that BI stands for 2. and if it is more than 2 to infinite, if the number is exceeding more than 2 to n number, which means you couldn't even count how many numbers they are producing, such kind of the fission process that happens is called multiple fission. Again telling you students, what is meant by fission? Fission is a process of splitting of a mother cell into daughter cell. Is if that daughter cell produced is exactly 2 in number, then that fission is binary fission. If the cell produced is more than 2 to infinity, which means it is not easy to count even to that extent, if an organism is doing the fission, then that kind of fission is simply called as multiple fission. Is so now let me make the diagram for what the binary fission is. So now binary fission, it's given the two organisms here, one is amoeba, the other one is leishmania. Is here it's given the diagram only for amoeba. Yes, we know that amoeba is a protozoa, which means it is of irregular shape. But leishmania, it exactly looks like euglena. It looks like, don't think that it's euglena. Yes, this is a leishmania. So amoeba, first step what it will do is, it will split its nucleus. Yes, leishmania too, it starts splitting up its nucleus. And this is the flagella for that too. Even the flagella it splits. And the third step is the cytoplasm starts splitting into two, and the nucleus, you see, it has moved to its respective positions. And here the leishmania, and now what happens? The groove that starts developing is making the groove to develop, and later the leishmania completes its fission process, and here amoeba, they are completing its fission process to form into two individual daughter cells. Yes, so this is all between the transverse and the longitudinal cut that happens between this organism. But you see the common between this amoeba and leishmania is both they are participating in binary fission process, but the way how it divides the cell, we are differentiating that into two. And this leishmania is a what? Is a major thing that causes a disease called Kala Azar. Yes, it's a frequently asked 1 mark question here. That's all about your binary fission. And next one, what we are going to see here is the multiple fission. I told you what is multiple fission. Multiple, which means multiple fission. Here, unlike the binary fission, here the number of cell produced is more in number, but there it's exactly 2. So here the organism, what we are going to take it for example is Plasmodium. Plasmodium is a protozoa that causes a disease called malaria. Yes, malaria is a disease that caused by an pathogen called Plasmodium, which is a protozoa. Yes, so don't tell that malaria is caused by mosquito. No, mosquito is a vector that carries this Plasmodium from infected person to a healthy person when they are biting a new healthy one. Yes. So this Plasmodium, they possess a nucleus and they have its own cytoplasm around, and once the Plasmodium they starts doing its multiple fission, so the nucleus splits into many fragments. Yes, the nucleus splits into many fragments, and then later the nucleus gets surrounded by its own cytoplasm of the mother cell. Yes, I guess now it's very clear for you. Now you see the partitioning of cytoplasm starts to occur. Yes, as you can draw many cells here, but time is a limiting factor here, so that's the reason I'm completing here with this. So this one is the thick cell wall of this organism. Yes, once that all completed this process, then next is the rupturing occur. Yes, so that the organism produced will be released. So now as a result of fission process, the organism produced is more in number. So again telling you, fission is of two types based on how many the daughter cells are produced in every fission process. Is number one is if the number of cell produced as a result of fission is exactly 2, then that fission is binary fission. If the process of fission during which the organism produce n number, n is in the sense more than 2 to infinite, that may be 1000 billion whatever so the number is, if that happens, then that fission is multiple fission. Is it's all about your binary fission and multiple fission. Is next one is regeneration. You know what is meant by generation? Generation in the sense to produce. To produce or to make. Re in the sense again. So regeneration is a process of asexual reproduction where the organism will get broken into several fragments. Yes, and each fragment, whatever the parts it got missed during the fragmentation, will start producing its own missed parts and making a complete framework for that organisms. Okay, என்ன அப்படினா துண்டு துண்டா அந்த ஆர்கானிசம் பிஞ்சு போயிடும். பிஞ்சு போன உடனே என்னென்ன பார்ட்லாம் மிஸ்ஸிங்கா இருக்கோ அந்த பார்ட்லாம் அது ரீடெவலப் பண்ணிக்கும். That is called regeneration. Is so for here the example given is planaria. Yes, planaria is the organism that we are going to study as a model organism for regeneration. Yes, let me make the diagram for planaria here. So this is the planaria. Assume that this is head and this is middle piece and this is tail. Yes, assume that we are going to dissect this organism into 3 parts. Yes, now I am going to make a transverse cut with this organism. Yes, now as a result of this cut, that is this fragment is this organism is getting fragmented into 3 parts. Yes, so now you see this end up in getting like this structure. And this one is the tail portion, isn't it? So now you see for the piece 1, what are all the parts to be regenerated? That is the middle piece and the tail. For the middle piece, what are all the parts is missing here is the head and the tail. For the tail piece, what are all the missing parts here are the middle piece and the head portion. So now this organism, they regenerated this part and they made into an entire organism, and this organism here, this middle piece, they regenerated the head and the tail, and this part has regenerated the missed portion of middle piece and the head. So now it's very clear that regeneration is a process through which the organism has the tendency or the ability to generate the missed out parts. Yes, what makes here the regeneration to work efficiently? So now you see here, this is the place and this is the place where the cell they may start dividing. Yes, making the missed out organ. So they are completing up with their own framework. Yes, so this is the best example for regeneration. And one more the organism here given but not in the diagrammatic manner is the hydra. Hydra too, they generate their missed out organ. Is say for example This is the structure of your hydra, and these are the tentacles. Yes, and this is the, this is the place through which the food can be intaken as well as the excretory substance can be ejaculated out. So if I make a longitudinal cut in the hydra, yes, so what you expect here is, this is piece number 1 and this is piece number 2. Yes, and as a result of regeneration, what here, this is the missed portion that got regenerated, and what here is the missed portion is this part, and they complete the regeneration step here. So now you see, as a result of regeneration, the mother hydra, they got fragmented and they made their own Xerox copying of making the 2 new organisms out of this process. Yes, the next one is budding. Here we are going to see budding in 2 organisms. One is yeast, the other one is hydra. You know where this hydra lives? Hydra is aquatic in habitat. Yes, so now let us focus how this yeast they do the budding process. And you know that yeast is widely used in baking industry, and yeast is the unicellular fungi. Yeast is the unicellular fungi. Most of the fungi they are multicellular, exception is yeast. So this you can expect a very often in GK question. Yes, so yeast is the unicellular fungi, and this is the structure of the yeast cell. And now once you give yeast more nutrition, they start developing a bud on its surface. This is the bud. Okay, a small bud that gradually they starts to develop. Yes, fine. So later on, the bud, it keeps proliferating and they start becoming big in size, and once they reach the considerable size, this new bud, they may start making a new bud and it keeps growing tremendously. Yes. So in today's class, we have discussed about what is sexual reproduction, what is asexual reproduction, and why DNA copying is very, very important and why it is not accurate. In the next forthcoming session, we shall continue with the topic that what already I told you in the previous class. Is thank you students. Thank you once again.